Welcome to Art Biolab. In the world of science, we often focus on the big breakthroughs. But behind every discovery, there's a world of meticulous, crucial work that makes it all possible. Today, we're going to pull back the curtain on one of these essential processes, the high-stakes detective work of cell validation.
Just imagine this for a second. A research team spends years, maybe even a decade, developing a revolutionary treatment for a disease. And then, the unthinkable happens. They discover they were working with the wrong cells the entire time. Now, this isn't some science fiction plot. It's a very real problem called cell line cross-contamination. It's happened before, and it can derail entire fields of research. It's truly a case of mistaken identity that can poison science at its very core.
So how do we guard against these cellular imposters? Well, that's what our explainer is all about today. We'll start by understanding the crisis itself. Then, we'll look at the forensic toolkits scientists use. And finally, we'll do a deep dive into one of the most powerful techniques of all, the art of karyotyping.
All right, let's get right into it. The absolute foundation of good science, of any reliable research, is knowing your subject. I mean, if you can't be 100% certain what you're working with, then every experiment, every result you get is built on shaky ground, just waiting to collapse.
So, what's the very first step to prevent this kind of disaster? You have to start with a trusted source. And in the world of cell biology, that means official cell banks. You can think of them as the Fort Knox for cells. They have incredibly strict protocols for testing and storage to guarantee that the cells you get are exactly what the label says they are.
But here's the thing. Getting cells from a reputable bank is just the beginning. The vigilance can't stop there. Over weeks and months of experiments, things can go wrong. So, throughout their research, scientists really have to become detectives, constantly re-verifying their cells' identities using a pretty impressive arsenal of forensic tools.
And the toolkit they have at their disposal is quite varied. They can analyze specific enzymes, they can look at unique protein markers on the cell's surface, or they can even do what amounts to a DNA fingerprint, very similar to what you'd see in a crime investigation. Each one of these methods offers a different piece of the puzzle to confirm a cell's true identity.
Now, you might be wondering, with all these options, how does a lab choose which one to use? Well, it often comes down to what that particular lab specializes in. You see, a biochemistry lab is already perfectly equipped to analyze enzymes. A genetics lab, on the other hand, deals with chromosomes every day, so karyotyping is a natural choice for them. It's all about using the tools and expertise you already have.
And that brings us to the main event for today, karyotyping. Now, this is one of the classic foundational techniques in genetics, but don't let its age fool you. It is still incredibly powerful. The best way to think of it is as the ultimate blueprint of a cell's genetic identity.
So, what exactly is a karyotype? Simply put, it's an organized picture of all the chromosomes from a single cell. Our chromosomes come in matching sets called homologous pairs, with one from each biological parent. A karyotype is what you get when you take a picture of all of them and then neatly arrange those pairs in order, from the largest to the smallest. It's like a definitive family portrait of the cell's chromosomes.
And this is where the power of the karyotype really shines. Just from this one single image, you can learn an enormous amount. It tells you the species instantly. Are these human cells, or did they get contaminated with mouse cells? It reveals the sex chromosomes, whether they are XX or XY. And, maybe most importantly, it can reveal large-scale genetic abnormalities, which is often a telltale sign that a cell has become cancerous.
So that's what it is. But how is it actually made? How do scientists go from a flask full of living cells to that perfectly organized chromosome map? Well, it's a fascinating and very precise multi-step process. Let's walk through how it's done.
The whole secret to a good karyotype is timing. You have to capture the chromosomes at the exact moment they are most condensed and easiest to see. And that perfect moment happens during a specific stage of cell division called metaphase. Scientists use a chemical that acts like a pause button, freezing all the cells right at this stage so their chromosomes are perfectly arranged for their close-up.
The entire lot protocol can really be boiled down to three main stages. First, you cultivate the cells, and then you arrest them in metaphase. Second, you treat them with a special hypotonic solution. This causes water to rush into the cells, making them swell up and gently pop, kind of like an overfilled water balloon. This releases all the chromosomes. And finally, you use a fixative to preserve that delicate spread of chromosomes perfectly on a microscope slide.
All right, so now we have our chromosomes neatly spread out and fixed on a slide. But at this point, they're basically invisible.
So the final part of the process is all about making them visible, making them give up their secrets. And to do that, scientists use special staining techniques.
There are two primary methods for this. The most common is called G-banding. It uses a dye that creates a unique pattern of light and dark bands on each chromosome. It looks a lot like a barcode, and you can see it with a regular light microscope. The other main method is Q-banding, which uses a fluorescent dye. This makes the chromosomes glow under a special microscope, revealing a different set of patterns. The method a lab chooses just depends on what specific details they're looking for.
So, regardless of how the chromosomes are stained, the final step is really an art form. It's like putting together a microscopic jigsaw puzzle. A high-resolution photo is taken of the chromosome spread, then a highly trained technician, either digitally or in some cases literally with a pair of scissors, will cut out each individual chromosome and painstakingly arrange them with their matching pair to create that final orderly karyotype.
And as our source material states very directly, this entire process is very time-consuming. This is not something that can be rushed. It's a craft that demands incredible patience, a sharp eye for detail, and immense dedication from these unsung heroes of the laboratory.
And that right there is why all of this is so profoundly important. This painstaking, time-consuming work isn't just about checking a box. It is the very bedrock of reliable, reproducible science. Without confirming the identity of these cells, every experiment that follows, every conclusion drawn, is fundamentally compromised.
The whole investigation really comes down to our most important witness, the cell itself, right? Every single conclusion, every little piece of data, it all rests on the testimony of this microscopic witness. And that means its identity has to be 100% certain.
And believe me, the stakes are huge. A reliable witness, the correct cell, gives you solid, reproducible data that can lead to some incredible breakthroughs. But an unreliable witness, an imposter cell, that contaminates the entire case. It's a massive waste of time, money, and it can send science down a total dead end. The consequences are just enormous.
So, where does this investigation even begin? Well, our first step is to check the witness's story by taking a close look at its behavior.
The first thing scientists look at is a cell's enzyme activity. You can think of this as the cell's job description. For instance, a liver cell's job is to produce a specific enzyme called arginase. If it's doing that job properly, that's a pretty good sign it is what it says it is.
Okay, but here's the catch. After a cell has been living in a lab culture for a while, away from its natural home in the body, it can start to, forget its original job. Its specific enzyme activity can drop off a cliff. It's like your star witness's memory is starting to fade, which makes their testimony a lot less reliable.
So scientists have a clever way to kind of jog the cell's memory. See, some enzyme behaviors are constitutive, which just means they happen naturally, all on their own. But others are inducible. You can actually coax them out with the right prompt. And this, my friends, is where the interrogation begins. The whole thing is basically a controlled interrogation. Scientists introduce a stimulus, maybe a hormone like dexamethasone or insulin, to prompt the cell. Then they watch closely for a reaction, looking for a spike in a particular enzyme's activity. If the cell responds the way it's supposed to, its identity just became that much clearer. But here's the thing. A cell's behavior is just one clue. For definitive, rock-solid proof of identity, investigators need to analyze the cell's unique biological signature, its fingerprints. This is done through something called isoenzyme analysis. And isoenzymes are just fascinating. They're enzymes that do the exact same job, but they have slightly different structures depending on the species or even the type of tissue they come from. These tiny structural differences create a totally unique pattern, a definitive genetic fingerprint for every single cell type.
And I mean it, just like a human fingerprint, no two cell lines have the exact same isoenzyme profile. Take the enzyme lactate dehydrogenase, for example. It has 5 different forms, and each one is found in a different type of tissue. By figuring out which form is there, you can pinpoint a cell's origin with incredible accuracy.
So what you get is basically a forensic report, just like this one. Investigators take an unknown sample and analyze its patterns for key enzymes like esterase and LDH. Then they compare that unique pattern to a database of known cell lines. And look at that, a perfect match between the evidence and cell line B. That confirms the sample came from a rat. Case closed.
So, how do they actually get these fingerprints? Well, it's a pretty neat 4-step process. First, they collect the evidence by cracking the cells open. That's called lysing.
Second, they separate the prints using electrophoresis, which sorts all the isoenzymes out by their structure. Third, they reveal the pattern by adding a substance that makes the different isoenzymes show up as visible bands. And finally, they make the match, analyzing that unique band pattern to confirm the cell's true identity, once and for all.
Now, all of this might seem incredibly detailed, maybe even a little over the top. But it brings us to the most important part of this whole investigation, why this level of certainty isn't just some nice to have, it's an absolute requirement.
Ultimately, the integrity of science really just rests on the quality of its evidence. Just like a court case can be thrown out because of tainted evidence, a scientific study can be completely invalidated by using the wrong cells. The entire search for truth depends on starting with a truthful witness.
So the key takeaways from all this can be summed up in this little code: trust but verify. That's the golden rule, right? It's why scientists can't just take a cell line at face value. Every cell has a story to tell, which we see in its unique isoenzyme fingerprint. Then there's the crime itself. Cross-contamination is what these methods are all designed to catch. And finally, because the stakes are so ridiculously high, certainty is the only acceptable verdict. This detective work is absolutely essential.
So this is basically the detective's toolkit. You've got your target, which is that unique marker on the cell. Then you have the detective, a special molecule called a monoclonal antibody. Now, think of this thing like a perfectly made key designed in a lab to fit only one specific lock, our target marker. And finally, you need a signal. This is usually a fluorescent tag that you stick onto the detective. So when the key finds its lock, a light literally turns on telling us, hey, here it is.
Okay, so how do we actually use this toolkit? Well, the first big technique is called immunofluorescence, and it's exactly what it sounds like. It's all about using that signal to make our target cells literally glow in the dark, giving us that undeniable visual proof that we found them.
Now, the simplest way to do this is called the direct method. Just think of it like sending in a detective who's already carrying a flashlight. You take your antibody, stick a fluorescent tag right on it, and then you just add it to your cells. It goes in, finds its target, latches on, and boom, the cell starts glowing when you put it under a special microscope. Super quick, super straightforward. But, there's also a more clever two-step approach called the indirect method. With this one, you first send in a plainclothes detective. That's an unlabeled primary antibody. It goes in, finds the target, and latches on. Then you send in the backup team, a secondary antibody, and this one is carrying the fluorescent flashlight. Now, this secondary antibody isn't looking for the cell. It's designed to find and attach to the first antibody. And here's the really brilliant part. You can get a whole bunch of these secondary antibodies to glom on to that one primary one, which makes the signal way, way brighter.
So, which one is better? Well, let's break it down. The direct method, it's simpler. It's just one step. But the indirect method is way more sensitive because of that signal amplification we just talked about. Plus, it turns out to be more cost-effective and a lot more flexible. And that's exactly why most labs end up using the indirect method for their detective work.
Okay, seeing a cell light up on a slide is cool and all, but what if you need to do more? Let's check out some advanced tools that take these exact same principles but apply them in some seriously powerful new ways, moving way beyond just taking a pretty picture.
So, we can adapt our detective work depending on the job. We've already talked about immunofluorescence, right? That's perfect for when you want to see where a cell is on a slide. But what if you need to count them or even sort them? That's where a machine called a flow cytometer comes in. Here, the cells are in a liquid, and they flow past a laser one by one. The machine counts every little flash of light, and, this is the amazing part, it can actually physically separate the glowing cells from the non-glowing ones into a super pure sample. Then, you've got immunocytochemistry. It's kind of like our glowing technique, but instead of a flash of light, it uses an enzyme to leave a permanent colored stain. That's perfect for looking at tissue slices under a regular old microscope.
Okay, so looking at surface markers is a fantastic tool, but for the absolute, undeniable, 100% proof of a cell's identity, for that, we have to go deeper. We need to look at its fundamental genetic code. We have to look at its DNA.
And this really gets to the heart of why this matters so much. DNA fingerprinting is the ultimate form of quality control. It's what's used to validate entire cell banks and to catch the #1 villain in cell research, cross-contamination. That's when one type of cell accidentally gets mixed in with another, and believe me, it can make an entire study completely worthless.
So how does this actually work? Well, think of a cell's DNA like a gigantic book. Scientists don't have to read the whole thing. Instead, they use a technique called multiplex PCR to basically photocopy very specific, short sections. These are known as short tandem repeats, or STRs. You could think of them like little genetic stutters, short sequences that repeat over and over, but the number of repeats is different for everyone. By looking at a few of these spots, you can create a unique DNA profile, a genetic barcode that's so precise it can even tell apart different cell lines from the same species. It really is the ultimate cellular ID.
So, from a simple glowing marker on a cell's surface all the way down to its unique genetic code, our ability to identify cells is just incredibly precise now. This is what lets us do things like purify stem cells, diagnose diseases, and protect the integrity of science itself. Which leaves us with a pretty fascinating question to think about. Now that we have this incredible power to find any cell we want, what's the next big mystery we should be trying to solve?